Hi, good day everyone. My name is May Castro and I am a grade 12 student from section Bernoulli. So we're here today because I'm gonna demonstrate to you the steps in the home experiment that I am going to do, which is called DNA extraction. But first, let me read the objective of this experiment to you. So the objective of this experiment is to demonstrate the initial step of genetic recombination, which is extracting of DNA. And for today's instance, we will be using the DNA of this banana. So here are the materials needed for this experiment. So we will be using ripe bananas, a Ziploc bag, a tablespoon, clear glasses, toothpick, salt, cheesecloth, a strainer, a dishwashing liquid, and of course the cold alcohol. So here are the total of our materials. In addition, we're also going to use a chopping board and a knife for cutting the bananas, but this is also both optional. Prepare the solution by mixing water, salt, and dishwashing liquid. We will need at least a cup of water, a tablespoon of salt, and a tablespoon of dishwashing liquid. This is very approximate, so don't worry if you don't get the exact proportions that we are using here. Place your isopropyl alcohol into the fridge or to a bucket of ice water to chill. So it is important that your alcohol is cold because it allows us a greater amount of DNA to be extracted. Otherwise, if it is warm, it may cause the DNA to break down and you definitely don't want that. We have to peel the bananas and cut them into small pieces. We use 2 pieces of banana since they are small, but you can also use 1. It will depend on how much DNA you want to extract. After cutting the bananas, you can place them in a Ziploc bag. After placing the sliced bananas in the Ziploc bag, pour the solution on it. Then make sure that you seal the bag and press out all the air. Next, we're going to mash the banana with the solution we made. You can mash it with your hands very carefully or use the back of the knife. Either way, make sure you end up with a very smooth consistency. Make sure to take your time and spend a few minutes doing this. Strain the banana mixture. You can use either a filter paper or cheesecloth to filter the mix. Use a clear glass so you can see it well. The DNA will be dissolved in the water that pours through, so now you know why we added water in the solution. This will take a few minutes, but after a while, you should end up with a crystalline liquid or semi-crystalline, depending on how thin your cheesecloth or filter paper is. Transfer liquid into a small container. If you have a test tube, you can use that, but if none, you can use any clear glass that you have at home, just like what I am using here. Pour the alcohol into the mixture slowly. This alcohol has been in the fridge. We have to slowly pour it on top of the mixture because you don't want it to mix with the banana mixture. We just have to make a fine layer of alcohol on top of it to help the DNA separate from the rest of the banana. Can you see that white slime forming in the middle of the alcohol layer? That is the banana DNA. Take note that DNA doesn't dissolve in alcohol, so it will help the DNA clump together in a separate layer. Now, just for the fun of it, let's try to fish out some of the DNA. Take a toothpick or any wooden stick. Try to swirl it and see if you can get any of the DNA out. Since DNA is such a long molecule, the DNA of one cell gets entangled with the DNA of the other cells, such that it is very easy to pull them out. Here is the DNA of the banana. I am glad that we performed the experiment well and successfully extracted the DNA from it. So there you have it, mga kabayo. So what you have seen today is not a magic. It is what science. So what is DNA? So DNA, or deoxyribonucleic acid, is a molecule that contains the genetic code of all organisms. This can be seen in animals, plants, archaea, protists, and bacteria. So in simple terms, it is a special molecules that can be found in the cell of all organisms, and it contains the instruction manual on how to be used. Why did we use banana as our sample? Actually, you can use other fruits too, like strawberries and kiwis. But we use bananas because they are triploid. This means that they have a lot of DNA inside of their cells and we can extract a lot from them. Another reason is because bananas can be easily mashed up. They have weaker cell walls and nuclear membranes that allow the DNA in the nucleus to come out more easily when placed under physical pressure. Why did we mix liquid detergent in making our solution? The outer skin of the banana or the membranes that holds the cells together in the banana are made of lipids. When we say lipids, it is actually the fats and oils. We already disrupted the outer membrane of the banana by crushing it, but we still need to further break down the cell and nuclear membranes. In doing so, we will use liquid detergent since we know that liquid detergent cuts through the grease. In short, it breaks down fats. Why did we add salt in making our solution? The salt will help neutralize the DNA charge. It will also make the molecules less hydrophobic, meaning it becomes less soluble in water. And since the DNA is tangled up with all the proteins in banana, the salt will help remove the proteins bound to the DNA and keep the proteins dissolved in water. So, once the dish soap helps release the DNA, the salts will help the DNA strands to stick to each other in clumps large enough for you to see. Why did we slowly pour the alcohol into the mixture? We slowly pour the alcohol because we don't want it to mix with the banana mixture. DNA is insoluble in alcohol, so specifically in cold isopropyl alcohol. When we say insoluble, it means it do not dissolve in alcohol. So the moment that we add our alcohol into the banana mixture, the DNA separate from the rest of the banana mixture and it forms a cloudy formation or it forms a clumps, just like what we saw earlier, and enable us to extract the DNA successfully. So that's the end for this experiment. I hope that you enjoyed watching it as well as learned something from the experiment that we did. Keep safe and God bless everyone.
Banana!